Now let's get on to the main reason we're here tonight, and that's to listen to Erica Sonnenberg, who is going to be lecturing about the microbiome and health. Erica is a senior research scientist at Stanford University School of Medicine in the Department of Microbiology and Immunology. She studies the role of diet on the human intestinal microbiota. She has published her groundbreaking scientific findings in prestigious journals such as Cell, Science, and Nature. And she's also the co-author of a great book, The Good Gut, Taking Control of Your Weight, Your Mood, and Your Long-Term Health. I can't think of a better topic to have right now as we're all struggling with the changes in our lives with the pandemic. So thank you again to all the participants, and thanks to Erica. Welcome.
Okay, thanks so much for inviting me and giving me this opportunity to speak. And I'm hopeful that in the next 45 minutes or so, you guys can get a break from listening to lots of conversations that we've had recently about viruses, and instead talk about my favorite type of microbe, which are bacteria. And I'm going to talk to you about some of the studies that we've been doing in our lab here at Stanford, understanding how we can use diet as a lever to improve our microbiota and our health. So I want to start with a picture of the microbiota or microbiome. I use both terms interchangeably so that we can get a visual on what exactly we're talking about. So in this image here, in the top left, are all the bacteria that are residing in a colon. And these are all the little tiny jelly bean looking things that you see here. Those are all different types of bacteria. And in the bottom right of this image, those blue circles, those are nuclei of intestinal cells. So this is where the intestinal wall sits. And this green stripe that you see through the middle is the mucus layer that our intestinal cells secrete. And it's sort of like a fence that we've created. And we like to think of this fence in terms of the expression, good fences make good neighbors. If that fence is in good shape, then our microbes stay where they're supposed to stay within the lumen or the inside of our colon, versus when the intestinal mucus layer thins, and there's concern that microbes can then penetrate that wall and get inside of our tissue, which is not where we want these microbes to be. So what we've learned over the past 20 years of really understanding more completely about our gut microbiota is that humans are in fact a walking ecosystem. We harbor trillions of microbes. In fact, close to 100 trillion reside just within our gut. And I hope that this image really gives you kind of a profound understanding of just how integrated these microbes are to our biology. They are sitting right next to our cells. There's lots of them, and they are as integrated into our biology as our heart would be, or liver, or any other organ. If we add up the total number of genes that the microbiota harbors, it's actually 100 times more genes than our own human genome. So if you want to think about yourself in sort of a genetic standpoint, from a genetic standpoint, we're actually more like 99% bacterial and only 1% human. And in my talk today, I'm going to talk about the promise of the microbiome as a therapeutic target, the connection between what all of us in the industrialized world harbor, this industrialized microbiome, and how we think that this is connected to a lot of diseases that we see rising in the West. And ask the question, can we modulate the microbiome in humans using diet? Diet is a great way to influence the microbiota because it is a change that we can make tomorrow. We don't have to wait for FDA approvals or long, expensive trials. So we're hoping that diet can be an important lever in controlling this community. And then finally, I'm going to end with some challenges that we're facing in maintaining a healthy microbiome. Okay, so as I've said, the microbiome is integrated into all aspects of human biology. Over the past 20 years, we've learned that this collection of bacteria don't just help us digest our food, they're actually integrated into our metabolism. So the calories that you're going to eat at dinner tonight, how many of those calories are stored as fat versus burned, is largely dictated by the species of microbes that you harbor within your gut. Your immune system. So we know that the bacteria within our gut can help us fight off intestinal infections, but it's much broader than that. And that we understand now that the microbes within our gut can also dictate how long we have a respiratory infection or whether or not we have seasonal allergies. And then finally, it appears that our microbiome is actually wired into our central nervous system. And there's been connections found between disruptions in the microbiome and diseases such as autism, mood disorders, and other central nervous system diseases. So I want to tell you about an experiment that was done about 15 years ago that I think really opened up the eyes of the scientific community and the lay community that really demonstrated how profoundly influential these microbes are in our health. And this was an experiment that was done while I was a postdoc at Washington University in St. Louis. And in this experiment, there were both lean and obese mice. And the microbiota from the lean mice was transplanted into mice that didn't have a microbiota at all. We call these germ-free mice. And in a separate isolator, there were obese mice, and the microbiota from the obese mice were transplanted into also germ-free mice. So these mice either harbored a lean microbiome or an obese microbiome. Other than that, these mice were treated identically. They ate the same types of food, the same amount of food, drank the same amount of water, no difference in the amount that they exercised or slept. But what we found was that the microbiota, that the mice that received the obese microbiota actually increased their body fat twice as much as the mice that received the lean microbiome. And this is despite them eating the exact same amounts of food. So this experiment really demonstrated the importance of these microbes. And not only are these microbes integrated into our biology, but here was an example of microbes actually causing a disease, obesity, a disease that we know many of us in the West are struggling with. So to explain a little bit about how this is possible, how bacteria that live in our gut can influence things that don't seem to be connected to our gut, I'm going to give you a little overview of how exactly the microbiome works. So we think about our gut as being colonized by bacteria, and that is true for the entirety of our digestive system. So starting in our mouth all the way to our anus, there's bacteria. But the majority of these microbes actually live within our colon or our large intestine. That's where I showed you that image with the really dense population of bacteria. That's within the colon. And so when we eat foods that have complex carbohydrates or dietary fiber, something like broccoli, those complex carbohydrates actually transit most of the way to the colon, mostly undigested. And that's because it turns out the human genome doesn't really encode much capacity for degrading these complex carbohydrates. We're really bad at doing that. So these complex carbohydrates make it from our mouth all the way to our colon pretty much intact. And once they reach our colon, they serve as food for our microbiome. So our bacteria consume these complex carbohydrates. And while they consume them, they excrete their microbial metabolites, their sort of waste products. And some of these microbial metabolites can actually transit through our intestinal lining and reach our circulation. So these small molecules, these drug-like molecules, some of them, are actually circulating throughout our body at all times, and these are microbial in origin. And we know that that these molecules can influence things like our ability to control blood glucose, satiety, and can improve our metabolism and immune system. Unfortunately, unfortunately, in the industrialized world, we don't eat a diet that's very rich in complex carbohydrates. Our diet is actually rich in another type of carbohydrates, simple carbohydrates. So these are things like sucrose, starch, fructose, glucose. These simple sugars, it turns out, our human genome is very well adapted to degrade these simple carbohydrates. They get absorbed early in our digestion in our small intestine, and these sugars can then enter our circulation. And this is where we run into troubles with things like insulin resistance, metabolic dysfunction, and increased inflammation. And so I didn't tell you what happens to the microbiome when it's not fed a diet rich in complex carbohydrates. And we were interested, our lab ourselves, in understanding what happens if you eat this diet that's very rich in simple sugars and doesn't have a lot of the complex carbohydrates that the microbiota relies on. So we had two groups of mice, one in which we fed them a fiber-rich diet, and the other that we fed a fiber-deficient diet. We didn't actually feed these mice donuts and soda, although they probably would have loved it. But the nutritional component of the diet was very similar to a donut and soda diet. And then we asked the question, what happens to the microbiota of these mice? And we took pictures similar to the one that I showed you at the beginning of the talk. So this is a cross-section of the colon from these mice. And if you look at the mice that were eating the fiber-rich diet, you can see the bacteria are on the right side, the sort of red little dots there. Those are the microbes. The yellow that you see there is actually plant cell wall material. This is the complex carbohydrates that the bacteria are consuming. And then what we found was that this mucus layer was actually quite thick. This was the good fences make good neighbors. And the immune system of the mouse seemed to be doing fairly well. In the fiber deficient mice, however, we saw that this mucus layer was actually quite thinned relative to the fiber-rich diet mice. And what appeared to be happening was, in the absence of complex carbohydrates from the diet, the microbes in the gut were forced to rely on the only complex carbohydrates that they had access to, which is found in that mucus lining. So it turns out the mucus lining actually contains quite a bit of rich complex carbohydrates that the microbes can use as a backup carbon source when we don't eat a fiber-rich diet. So if you eat a fiber-deficient diet, as was happening in these mice, the microbes, in fact, start eating you. You become the food source now for these bacteria because you haven't provided the plant-based complex carbohydrates that these microbes rely on. In addition to this thinning mucus that we saw in the fiber-deficient diet mice, we saw a bloom in mucus-degrading bacteria. So those microbes really took over in this situation. And in the mice, we also saw increased markers of inflammation, an indication that this was not beneficial for the health of the mouse. We know in the West and throughout the industrialized world that inflammation-driven diseases are rising. Childhood obesity, which has always been quite high in Western countries, is actually rising fairly quickly globally as well. So this is turning into not just an industrialized problem, but as countries around the globe adopt our lifestyle, we're seeing the effects of that in terms of disease and specifically childhood obesity. We know that immune disorders are rising in the industrialized world as well. Autoimmune diseases like Crohn's disease, asthma, type 1 diabetes, all rising quite rapidly. And so if we understand that microbes influence our health profoundly, the question we can ask is, can we then manipulate these microbes in order to improve our health? If they have so much power over our biology, can we take some of that power back through understanding how they work and manipulating them in a way that benefits our health? And so one question that our lab at Stanford is very interested in answering is a very basic question. What is a healthy microbiome? What does a healthy microbiome look like? Because you can't solve problems that are rooted in a diseased microbiome if you don't understand what a healthy microbiome is supposed to look like. And unfortunately, for the past 20 years or so, much of the answer to this question has been answered using, in our opinion, a flawed model. And that model is, I think, perfectly illustrated by this Banksy art that he did. And the flawed model is that in the industrialized world, we have a human genome that is actually very similar to that of our Paleolithic or Neolithic ancestors. Our genome evolves quite slowly. You know, our generation time is about 20 years or so. The microbiome, on the other hand, adapts quite rapidly. Their generation time is about 20 minutes. So if you change what you're eating today, your microbiome will look different tomorrow. And that, from an evolutionary standpoint, was a very adaptable part of our biology. Here was an aspect of our biology that could really change rapidly depending on the food resources that we had at the time. Unfortunately, in the past, say, 50 to 100 years, our diet in the industrialized world has shifted towards that which is basically unprecedented in the history of humanity. And this is a diet that's very rich in processed, sanitized, calorie-dense, but nutrient-rich food. And so by studying the microbiome of Western individuals, these are people that are consuming this kind of diet, we worried that we weren't really going to adequately answer the question of what makes a healthy microbiome. Instead, what we were studying is, what does the microbiome look like in people that are healthy right now, but we know are predisposed to Western disease? And so instead, we wondered, could we find people around the world that hadn't made this shift in their diet yet? We're still consuming a diet similar to that of our pre-agrarian ancestors. And there are these people that exist around the world, and one such group of people are the Hadza of Tanzania. This is a group of hunter-gatherers that have a diet that's very similar to that of our Paleolithic and Neolithic ancestors. They don't farm, they don't store any food. So every morning they wake up and they go out and forage and hunt for what they're going to eat that day. And here's a group of Hadza that have foraged a bunch of tubers. So this isn't just a pile of sticks they're burning. There's actually tubers on here. So this is sort of the ancestral potato. And they're cooking these tubers, and this is actually their staple food. This is what they can rely on pretty much year round and what makes up the majority of their calories. And the tubers are foraged by mostly the women and children within the Hadza. And the men every day go out and they are the ones that are trying to get meat. So they hunt with bows and arrows that they make. Some days the hunts are successful and they will have a great meal of meat, but many days the hunt is not so successful and they subsist on basically a vegetarian diet. And so we wanted to understand what the microbiome of people like the Hadza look like. And so in this graph here, what I'm showing on the y-axis is the microbiome from people all around the globe, from Africa, South America, North America, Europe. And on the x-axis, what this graph shows is how similar those people's microbiota are to one another. So the closer the bars are to one another on this axis, the more similar their microbiota is. And what we found was traditional populations, regardless of whether those populations live in Africa or live in South America or Papua New Guinea, they all have a microbiota that's actually quite similar to one another and very distinct from that of people living an industrial lifestyle. So these are people in North America, Europe. Our microbiota all looks similar to one another and very distinct from that of these traditional populations. Okay, so what are the differences? Why are their microbiomes so different from ours? One, there are many things that differ. One of the most striking things is their microbiota is actually much more diverse than what we see in industrialized population. So they have many more types of species of bacteria living within their gut. So if you think of it like an ecosystem, theirs is like a lush rainforest. Ours is not so much. They also harbor species in their guts that we just don't ever see in Western populations. So the Western microbiota does appear to be deteriorated relative to the traditional population one. If I had to pick two images to describe how I see these two different microbiomes, the traditional microbiome would be this image here on the left, high diversity, high functioning, metabolic capacity of this microbiota. And their microbiota is very much tailored to the degradation of complex carbohydrates, likely from all the tubers and plant material that they eat. The Western or industrialized microbiota looks very deteriorated relatively. We're missing species, our diversity is low, and the capacity of our microbiota to degrade complex carbohydrate is also depleted relative to these traditional populations. So we've come up with this model for what we think has happened to the microbiota over our evolutionary past. Before 10,000 years ago, all humans obtained food through hunting and gathering. Agriculture is about 10, 20,000 years old. When agriculture was invented and humans shifted towards farming for most of their food, the microbiota had to adapt to that difference in food. And then about 400 years ago was when we started seeing the beginnings of industrialized food. And then just about 50 or 100 years ago was when processed and sanitized food became widely available in the industrialized world. And we think with each change in the way that humans acquired food, that the microbiota had to adapt, and that these adaptations has led to the deteriorated state of the microbiota that all of us in the West appear to be harboring. We actually did a mouse experiment to try to address this model specifically. And what we did was we took mice that harbor a human microbiome and we put them essentially on a processed, sanitized food diet. And what we found was with each generation of these mice, their microbiota became more deteriorated. They lost more species. And after 4 generations, these mice had actually lost almost 2/3 of the original diversity that the first generation of mice had. So this was a mouse experiment that indicated that diet alone is a very powerful lever in controlling the diversity of microbes that we have in our gut. And when we remove complex carbohydrates, we can really deteriorate the community that we have. Okay, so I've painted a pretty bleak picture here. This is the point in the talk that I'm sure a lot of people feel desperate, like there's just no hope. But I'm hoping that moving forward now, I can show you some data that provides a more hopeful view. We know that the microbiota in the West is deteriorated. We know that this deteriorated microbiota is likely causing or contributing to many of the diseases that are becoming so prevalent in the industrialized world. And there are probably many reasons why this has happened. I've talked about diet, but we know sanitation influences the microbiota, medications, formula feeding, all these things. So the question is, how can we get from this deteriorated microbiota that's contributing to disease towards a microbiota that's more healthful and beneficial? Is that just through changing diet? Are we going to have to reintroduce some of the lost microbes that these traditional populations have that we don't have? And so my husband and I have started this Center for Human Microbiome Studies, where we're trying to develop humans as a model for understanding the microbiome. Our lab has done a lot of mouse experiments, and those experiments are great in that we can really dissect what's happening to the microbiota and the host in a mouse model. But at the end of the day, they are mice. And what we really want to know is how we can influence in a positive manner the microbiome for humans. And so I'm going to talk about one of the early studies that we did with human participants. This was the fermented or fiber food study that we call FIFO. We took 36 healthy adults and we randomly assigned them to one of two diet groups. In one diet group, the participants were eating a high fiber diet of their own choosing. And in the second group, participants were told to eat as much fermented food as possible. We looked at a number of measurements for their microbiome and their immune system, both at the baseline before they started the study, in the ramp-up period where they slowly increased the amount of fiber or fermented foods that they were consuming, in the maintenance phase, so this was 6 weeks where they just ate as much fiber or as much fermented foods as possible. And then finally, this choice period where participants could either stick with their high fiber or high fermented food diet, go back to their pre-intervention diet, whatever they wanted to do. Okay, so can people do this? And the answer is yes. Human participants in studies are amazing. I'm used to working with mice that always do exactly what I tell them. So I was a little bit worried in working with a human population, but they did an amazing job. And you can see in the high fiber group here, participants on average were eating about 20 grams of dietary fiber at the beginning of the study. And then by the maintenance phase, they had almost doubled that, over 40 grams of dietary fiber. So amazing job there. This dotted line shows what the fermented food group did. So they were not told to increase their fiber, the amount of fiber in their diet, and they didn't do that. So that was great. In the fermented food group, participants at the beginning were eating about a half serving of fermented foods per day. That's pretty typical for the average American. But by the maintenance phase, they were eating around 7 servings of fermented foods per day, which was which was great. And then you can see here that the fiber group didn't increase their fermented food intake. So we really had two groups that had done this very surgical, precise intervention. One group eating a lot of dietary fiber, one group eating a lot of fermented foods. For the high fiber food group, we didn't want participants doing supplements. All the fiber had to be from foods, but we left it up to the participants how they wanted to increase their dietary fiber. Some ate a bunch of fruits and vegetables. Some really focused on grains and legumes. Everybody kind of did it in their own way, but we wanted to give them that ability to choose. And we just wanted to see more dietary fiber, doesn't matter where it comes from. Does that make a difference? I'm going to just give you a snapshot on how these people change their diet. So here's the participant at baseline, so before the intervention. It's a pretty typical American diet. You see a lot of processed food, a lot of eating out, fast food. This was very typical of our participants before. And then here's the same participant when they were doing the high fiber part of the diet. And you can see that they've switched their diet pretty significantly. Not only are they eating a lot of dietary fiber, they're not eating very much packaged food, if at all. And there's no, Starbucks or Domino's pizza in the high fiber group either. So these participants, with really the one instruction of eat as much dietary fiber as possible, really changed their diet dramatically. And so the nutritionist that was helping us run this study felt like this was amazing to witness. Giving participants one simple rule, eat a lot of dietary fiber, led to a bunch of other positive dietary choices. And in a time where it feels like there is new nutrition advice every other week, this can be a very simple way to keep your diet healthy without having to keep track of, am I eating enough fruits or vegetables? Am I eating too much saturated fat? If you focus on just eating as much dietary fiber as possible, all the other nutritional aspects seem to fall into place. We saw this cohort wide in this whole high fiber diet arm. And when we looked at the nutrient profile of what these people were eating, both at baseline and then during the maintenance phase, we found all these nutrients, vitamins, minerals, dietary fiber in green. These all went up when they were on the high fiber diet. So a lot of positive nutritional profile changes here. And we actually only saw two things go down, and that was sodium. So these participants were eating less salt, even though they weren't instructed to do that. That's probably a result of less processed food. And they were also eating less animal protein. So most of their protein during this study was plant-based. Okay, what happens to these people's microbiota? What we found was that participants in the high fiber diet, their microbiota shifted towards one that was better able to degrade complex carbohydrates from plants. So if we look at baseline in white, the maintenance phase, this is when they're eating the most fiber in green. And this is the amount of these complex carbohydrate degrading enzymes that we found in their microbiota. And all these guys go up when they're eating the high fiber diet. So this microbiota now looks more like the Hadza in that it is shifting its metabolic capacity towards that of degrading plant complex carbohydrates. We also saw that the small molecules that the microbiota was releasing into the gut looked beneficial as well. So these are some small molecules that are thought to be detrimental to human health. And what we found in the high fiber diet participants is when they were consuming the high fiber diet, these three molecules went down in abundance. So indicating that they had an improved metabolic profile within their microbiome. The other thing that happened to these people's microbiomes is they actually grew more bacteria. So we measured the amount of bacteria that was within these people's gut at baseline and then during the high fiber diet maintenance phase, and they actually had more bacteria. So indications of little seedlings in their ecosystem now sprouting and growing this more rich ecosystem. Okay, what about their immune system? What happened to that in the high fiber diet arm? That ended up being a little more complicated. So we had expected an overall decrease in inflammation in these participants, and instead what we found was 3 distinct responses. So here in this figure, each row here is an individual participant, and each column is a different measure of the participant's immune system and how it's functioning. And if in general, if the bar becomes more red, that's an indication that it's becoming slightly more inflammatory. And if it's becoming darker blue, then it's an indication that it's becoming less inflammatory. And we found that there were participants that in this one cluster whose inflammation appeared to get worse on the high fiber diet. And then these 2 other groups that in general had trends going down. And so we wanted to understand, why did some participants seem to have lowering inflammation on this high fiber diet, whereas others did not? And so we went back and looked at the microbiota of these participants at baseline before they started the intervention. And what we found was that the participants that had this sort of higher inflammation output were the participants that had lower microbiota diversity to start. So these were the participants that had sort of the most deteriorated microbiota of the group. And participants that had higher microbiota diversity at the beginning of the study were the ones that really benefited from the high fiber diet intervention. So this indicated that there were aspects of the immune response to the high fiber diet intervention that was not generalizable, but in fact participant specific. So we learned that high fiber foods improved the nutrient profile of the diet, shifted the microbiome towards more plant degrading capacity, improved the microbiota function and density. They had more bacteria than before the intervention, but the immune response to the intervention was individualized but related to the starting microbiota diversity that these participants had. Okay, let's shift over now to the fermented food group. So this was a group, in this arm, participants didn't have to change anything in their diet other than incorporating as much fermented foods as possible. They ate all kinds of stuff, kimchi, sauerkraut, yogurt, kombucha. They could do whatever they wanted for fermented food. Unlike the high fiber diet group, the fermented food arm, their nutrient profile didn't really change much. The only difference that we saw was an increase in the amount of animal protein they consumed. And this came from fermented dairy foods like yogurt that they were eating more of. We were concerned that perhaps participants' sodium intake was going to go up. A lot of fermented vegetables have a lot of salt in them, but in fact, we didn't see that. So as participants were consuming more salty fermented foods, they must have been cutting it out in other aspects of their diet, probably without even realizing it. Okay, what happens now to the microbiota of participants consuming fermented food? We saw a dramatic cohort-wide increase in microbiota diversity. These participants at the beginning of the study had about 100 different species that we could detect in their gut. By the end of the study, they had over 125 species. So they gained on average another 25% of microbiota diversity on top of where they started. And at first, when we saw these data, we thought, well, I guess that makes sense. I mean, they're eating bacteria, right? Fermented foods have live microbes in them. So this increase in species that we're detecting is just the bacteria that these people are consuming. And so we wanted to test that directly. So what we did was went out to the grocery store. We had food logs from all these participants, so we knew exactly what they were eating. We went out to the grocery store and we bought all the same foods that these people had consumed. We then struck these foods out onto these agar plates to see if there was actually live bacteria found within these foods. And turns out there were in every food. So when that label says live and active microbes, they mean it. There were live and active microbes in every fermented food that we tested. Then we went and identified which each of these strains of microbes that we detected in the fermented foods, what those microbes were. And this is the list that we have here in the middle of the different bacteria that we found in these foods. And for the most part, the microbes that we found in the foods were the microbes that were listed on the label. So in general, these fermented foods have live and active microbes, like they say, and are largely what is written on the label. So it turns out these are actually pretty high quality products. Okay, so what the 25 on average new species that we detected in these participants' microbiota, how many of those are the bacteria from the fermented food? When we looked at that, it turns out that this small sliver here, which is about 5% of the new species that we detected within the gut of these participants, came from the foods that they consumed. 95%, this whole purple part here, not from fermented foods. So it appears that the consumption of fermented foods is having an indirect effect on microbiota diversity. There's something about eating fermented foods that makes the microbiota receptive to new species. We don't know where these species came from, likely other parts of these people's environment, maybe from their homes, maybe from other people they're interacting with, but it didn't come from the fermented food. Okay, what about these participants' inflammation and immune system? In the fermented food group, we actually saw a cohort-wide decrease in many inflammatory cytokines. So here I have, I think, about 18 inflammatory cytokines. And over the course of the intervention, all these inflammatory cytokines decreased for the whole group. So unlike the fiber arm that had a more individualized response to the immune system, fermented food seems to have a powerful group-wide effect on decreasing inflammatory cytokines. So we learned that fermented foods increases microbiome diversity in participants and decreases inflammation. Now, every time I present this, people ask me, what about probiotics? Because fermented foods contain live bacteria. What if I don't like fermented foods or it's a pain to have them around the house? I just want to take a probiotic that has the live microbes. Isn't that going to be the same thing? So we wanted to test that directly. So this is another human intervention trial that we did. And in this one, we actually recruited participants with metabolic syndrome. We wanted to up the ante. We wanted to see not only can we change these participants' microbiome and immune system, but can we actually change the trajectory of their metabolic syndrome. This was a double-blind placebo-controlled study. So 14 of the participants were randomly assigned to a placebo group and 28 took a probiotic twice a day for 10 weeks. We have baseline, intervention, time points, washout, blood, stool, so we can measure their microbiota and their immune system during this intervention and ask the question, can probiotics improve participants' microbiome, decrease their inflammation, and reduce the severity of their metabolic syndrome? Okay, so we had high hopes for the probiotic because the fermented food did increase microbiome diversity. But in this study, we did not see an increase in microbiome diversity with probiotic consumption. So if you look, the probiotic in purple, the placebo in gray, it's all the same from baseline to the end of the study. So no change in microbiome diversity with the probiotics. What about inflammatory cytokines that we saw decrease so dramatically in the fermented food? Did that work for probiotics? And the answer, unfortunately, was no. No difference between the probiotic or the placebo group, and also no difference over the course of the study either. So inflammatory cytokines stayed the same throughout the study. Okay, what about markers of their metabolic syndrome? We looked at insulin, glucose, lipids, all those things unchanged during the study. So at this point in this was a pretty depressing point in the study. We had done this intervention. It was complicated. It was expensive. We were hoping to see changes in the microbiota or immune system or anything. We were seeing absolutely nothing. And so we thought, okay, before we just write up this negative result, let's take a closer look at individual participants and see if there's any information we can get from that. And so here what I'm showing in this heat map is each participant is a row here. And so this is the placebo group. This is the group that took the probiotic. And then each column is a different measurement we took of their sort of a metabolic syndrome symptom. And what we did was basically feed this into a computer and ask the computer, do all the probiotic participants look the same? Or do you see anything, any groupings within the probiotic group that are different from one another? And what the computer spit out was actually 2 different groups that it identified. In one group, shown here in the middle, there was really not much of a change. So that's indicated by the sort of white and light blue in most of their markers. But in some cases, there were actually worsening markers. So worsening triglycerides here and insulin here. That's when it goes red. But in this group down here, we actually saw improvements. So that's indicated by the dark blue. And these were statistically significant improvements in their triglycerides and their insulin. And so we termed this group responders. So these were people that seemed to be getting a benefit from the probiotic intervention. And so we asked the question, why? Why are these people benefiting from the probiotic intervention? And the first place we looked is their baseline microbiota composition wasn't different. Baseline microbiota diversity wasn't different. What about their immune status? Maybe there was something about their immune system that was different at baseline between these 2 groups. No difference. At this point, we remembered that in addition to this intervention, we asked participants to keep food logs, write down what you're eating throughout the course of the experiment. And when we went back and analyzed these food logs, what we found was that the responders actually had a diet that was different from that of the non-responders. And specifically, these responders were eating more vitamins and minerals and had a slight increase in their dietary fiber consumption. But in general, what we observed was that the responders were eating more plants in their diet than the non-responders. So this was an indication that to gain the full benefit of this probiotic, you had to be eating a diet that was more rich in plant complex carbohydrates. So what these studies taught us is that fiber and fermented foods do different things to our microbiota and our immune system. So if we go back to our broccoli example, when you eat a high fiber diet, those complex carbohydrates make it to the colon where the majority of our gut bacteria live, and these gut bacteria consume these complex carbohydrates. That resulted in participants having a denser community of microbes within their gut, better function of those microbes, more capacity for plant carbohydrate degradation. And we also saw lower inflammation if participants had a higher baseline microbiota diversity. In the fermented food group, what we found was that the consumption of fermented foods led to a higher diversity of their microbiota and lower inflammation. And for probiotics, what we found was that probiotics appeared to work best in the context of a better diet. So these data really point to a diet that blends both high fiber and fermented foods as having the likeliest, most beneficial impact on the microbiota and the immune system. So here I have some examples of blending high fiber and fermented foods, yogurt with nuts and berry, kimchi fried rice, which is a favorite in my house. So just different ways that you can incorporate high fiber foods and fermented foods we think would have the most benefit. So at this point in the presentation, I'm sure you're all thinking, okay, I'm convinced. Tomorrow I am going on a high fiber, high fermented food diet. If Erica can just give this talk to a bunch more people, then we can reverse trends of inflammatory diseases and global obesity trends and problem solved, right? But I'm going to talk to you now about why This is a lot easier to explain than to solve. When we did our study on the microbiome of the Hadza hunter-gatherers, this was published in Science, and the journal decided to feature our article on the cover. And they chose this photo, which is a photo of a Hadza gentleman holding a piece of honey. For this man to get honey, what he had to do was hammer pegs into trees, because most of these hives are high up in trees. He had to start a fire on some twigs. He had to climb those pegs, holding his branches of fire, so that he could then smoke the bees out and grab the honeycomb. And you can see actually in this picture here, his finger's swollen. So he's getting stung this whole time. And Hadza have actually died from this endeavor. They get up there and, there's bees everywhere and they fall. And so everything that this man had to go through to get this honey, if we had to do that every time we wanted a Frappuccino or a donut or something like that, we would eat way less of that food. And so what has happened through our evolutionary history is, because before processed sanitized food existed, getting calorie dense things like honey or meat required risking our lives. We had to climb trees. We had to go out and hunt with bows and arrows. And so our brain had to really reward us for that, because these were dangerous endeavors. If you ask the Hadza what their least favorite food to consume is, it's those tubers I showed you. They hate those tubers, but that is the majority of their diet, because it's easy to get and they don't have to risk their lives. So these Hadza are eating a high fiber diet, not by choice. This is what's available and easy for them. If you brought these individuals to New York City and showed them a McDonald's or whatever, they would be making all the terrible food choices that we make, because we are inherently wired to crave these dense, calorically dense foods. And so it raises this question of, how can we make good food choices when in the industrialized world, we essentially live in a convenience food environment where calorie dense, nutrient poor food is readily available and inexpensive. And there are probably many ways to answer this question. Some of it is probably stems around education and more research, changing policy so that these types of convenience junk food are actually more expensive than healthier foods. Maybe it's some answers are in food technology, making what is inherently unhealthy food but tastes good more healthy. Somehow can we, insert dietary fiber or other healthful nutritional components into sort of junky food. And then the final thing that I have on here is hacks. And these are just like ideas that I have used in my family to try to get us to eat healthier. I have 2 teenage daughters that, it's hard when you're a teenager to think about things like concerning yourself with, am I going to develop diabetes 20 years from now? Am I going to develop some autoimmune disease down the road? And so these hacks are ways that I've come up with for trying to make our family make healthier choices, and doing that in a way that's as easy as possible. So these are things like not buying foods in the grocery store that I wouldn't want to eat. So that means like minimizing the number of packaged foods that I buy. The other trick that I like to do is, my family really likes ice cream, and I try to make it a rule not to buy ice cream when I go to the grocery store. So after dinner, if we're all sitting around and we're like, oh, ice cream sounds really good, because I won't buy it at the grocery store, what that involves is us getting in the car and driving to a place and getting ice cream. I'm trying to like recreate the climbing up the tree and smoking out the bees, making it hard to get those kinds of food. And some nights we decide we are up for it. We're going to get in the car. We're going to go get ice cream. But most nights we're just like, never mind. Instead, I'll have an apple for dessert. So I just want to throw that out there and get people thinking about ways that we can try to trick our brains into choosing more healthful choices. And so finally, I'll just finish by acknowledging the members of our lab that have done much of the work that I've shown you today, and then our various funding sources. And then, as mentioned, my husband and I wrote this book, The Good Gut. So if you're interested in learning more about the microbiota and how it relates to health and disease, I suggest that you take a look at that book. And thank you so much. And I'm happy to answer questions that people have.
Well, Erica, that was absolutely terrific. You got to be able to hear someone clap. That was truly phenomenal. Walk through the science of the microbiome, and then importantly, some of the important interventions that you're trying to explore that could really help change the microbiome. And being a cardiovascular outcomes clinical trialist, I certainly hope at some point in time you're going to be able to show true impact on important diseases as you further understand what kind of needs to be impacted in the microbiome to see those important outcomes. So doesn't surprise me from the Project Baseline Health Study participants. We have lots of great questions, and we have about 10 or 11 minutes to go through some of these. So I'm going to ask these to you in kind of rapid succession, and maybe we could have, as brief of answers as you think you can provide, so we can get through many of them. But if you need to go into much more detail to help explain it further, of course, take your time. So the first one, and very appropriate with the pandemic, how does sheltering in place and limiting contact with the world beyond your home or bubble impact your microbiome? And if so, anything you recommend?
Yeah, so, I've thought a lot about that. I talked a lot about diet and the influence of the Western diet on our microbiome, but another thing that we know influences the microbiome is sanitation. And one of the differences between these traditional populations and industrial populations is that we just have so much more sanitation here than they have. And it's clear that exposure to environmental microbes is something that our biology expects, because for our evolutionary past, this was something that was just a part of our daily life. And when this pandemic hit and I saw hand sanitizer flying off the shelf, the first thing I thought of was like, this is great for right now, but my worry is that we're going to even go further into being more sanitized than we already are. And we know that there's a cost associated with that. Now, of course, we don't want to go back to drinking dirty water or not washing our hands, because the risk of exposure to pathogenic microbes is too high. So the question is, how can we expose ourselves to benign, beneficial, healthy microbes? And I think the answer for that really is fermented foods. These fermented foods are teeming with bacteria. They speak to our immune system. They're interacting with our microbes. And these are a safe way to expose ourselves to bacteria at a time when we are, Clorox wiping everything. So I would just encourage people to find safe ways to interact with environmental microbes. And that can be consuming fermented foods. That could be, gardening projects in your yard where you're not spraying pesticides or insecticides and you're getting dirt on your hands, and maybe you're not being so careful about washing your hands. We have a dog in our house. When he just is hanging out in the backyard and we're petting him, I don't worry about washing my hands before I eat or put my hands near my mouth. And that's just a way of trying to get microbes sort of back into our system.
That's great. You answered this in part, but it's about probiotics. There's a lot of them on the market. And you have some data now showing that maybe in some people, if you modify your diet a bit, you could be a responder with a probiotic. Do you have enough information yet to say to people, if you want to change your diet and you want to take probiotics, that might be an okay thing to do? Or do you think more work is needed?
I mean, I think more work is needed if we want to really make precision recommendations. So ideally, the situation would be we would understand enough about these probiotics and how they interact with a person's specific microbiome in a way that we could say, here's what your microbiome looks like, here's the disease we're looking to treat, here's the probiotic that we know through studies will benefit you. Unfortunately, we don't have that data yet, although I think moving forward, as more of these studies come out, we will eventually get that data. But it's clear that there are benefits for probiotics. But at this point, it's sort of, we have to be kind of experimentalists and try them and see, I'm going to try this probiotic A. Do I feel better? Am I having more regular bowel movements? Do I notice my allergies seem to be getting better? And if that's not the case, then maybe a different formulation of probiotic would help, or changing other aspects like this, putting probiotics along with trying to improve your diet might help. But ideally, we would have concrete recommendations, but we just aren't there yet.
And did you look at the group of patients that perhaps didn't respond? Because the overall population didn't have a response. You found a group of people that were responders. That means that there had to be a group of people that were actually went in the wrong direction. They got worse. Were you able to look at that group yet, or is that ongoing?
That is ongoing. Unfortunately, our study was quite small, so that probiotic cohort was only 28 people. And then when you divide that in half, now you only have 14 people to do statistics on. So we see some maybe tantalizing stuff, but a lot of it doesn't hold up to the statistical tests that we do. So we're hopeful in the future that we can use this as a pilot and do a much larger study and really get at those the answers to those questions.
That's great. I think this is a quick question. If you become a vegetarian or vegan, would your microbiota change in weeks, months, or years?
Yeah, so I mean, I hope what I've been able to get across is that the microbiome is quite responsive to our diet and our environment. And so if you change your diet tomorrow, like literally tomorrow, your microbiota will change. Those changes are small. And so what we see is that while the microbiota is quite responsive, it does have this tendency to revert back to the community that you had prior to your changing in diet. And we think that this actually may be part of the reason why weight loss is so difficult. You get people to, people can change their diet, lose weight, but there's this force that seems to be bringing them back to their pre-diet weight. And we see when we look at microbiomes of people that have lost weight, their microbiome is also shifting back to their pre-diet form. And so while the microbiota is responsive on a short term, if you really want to have long lasting changes to your microbiota, you need to sustain those dietary changes over long periods of time. And we don't know how long it takes. So, if you're I eat meat every day kind of person, and tomorrow you become a vegetarian, it may take your microbiome several years to sort of forget the meat eating state that it was in and really entrench in your new sort of vegetarian microbiome. So the answer is actually kind of, two different time scales.
Yeah, no, that's the tough part. It's the sustainability of any kind of intervention over time. I tell the story, many have heard it already. My wife was a vegetarian before she met her cardiologist, meat-loving husband of the future, and then now she eats meat as well. So it is hard to sustain that over time. Does the gut microbiome exclusively contain bacteria, or does it also include fungi and other viruses?
It's all there. Fungi and viruses are there. Most people, when they talk about the gut microbiome, are talking about bacteria, and that's just because that's mostly been what we've studied. But there are actually even virus. So, bacteria have their own viruses. Those are called bacterial phages. And those are in the microbiome as well. And so, these bacteria really have it tough in our gut. Like they're trying to survive on the food we're eating. And then they're dealing with their own viruses that are in there too. So it really is like an ecosystem. There are predator-prey interactions going on. And there is, basically every microbe under the sun is there.
Fantastic. Let's do maybe 2 more questions and be respectful of people's time. Statin drugs, commonly used in people with and without cardiovascular disease. Does it affect the microbiome?
Yes. So there is evidence that these drugs, like many drugs, influence the composition of people's microbiota. And that's the thing I think the past decade of research has really brought to light is just, we have to start thinking about these microbes as an integral part of our biology. So if you shift one aspect of your biology through drugs or whatever, that has a reverberation on the microbes in the gut. So the microbiota is sensitive to these kinds of things. And statins and metformin are 2 drugs that do influence the community.
That's great. It's the intersection, right, of the microbiome, inflammation, metabolic syndrome, cardiovascular disease, many cancers now, and other chronic conditions. So it's really, our understanding of this has evolved tremendously. Mm-hmm. Last question, hopefully not a complex one, but how can I know if my microbiome is optimal or not?
That's a tough one. I think, I think a few years ago, before we were studying these traditional populations, we had a sense of that based on studies of healthy Westerners. I think now these traditional populations making me question everybody's microbiota in the West and how healthful it really is. What I tell people is at this point, there's no test. We were hoping there would be this like list of bad bacteria and list of good bacteria, and you could just kind of tally up what you had. That doesn't appear to be the case. This is a complex ecosystem. And so it's not just which microbes are there, but what they're doing and how they're interacting with the other microbes that are important. People just need to, I say, pay attention to your bowel movements. You want those to be regular. You want those to be, passing easily. That's an indication that you have a microbiota community that's doing well. Other than that, we're just sort of like still trying to figure out what a healthy microbiome looks like. And it'll probably be different depending on your age, depending on, other things happening in your life. And so there won't be one simple healthy microbiome. There'll be probably many configurations.
Well, thanks. I guess it's a good plug to get those stool samples sent in once we restart the visits again so we can start studying this in larger populations.
Yeah, absolutely.
Erica, thank you again. Absolutely terrific presentation. Many comments on the internet saying it was a phenomenal talk and much appreciated. So thanks for joining us this evening. Appreciate it.
Thanks so
much. And thanks to you all. Thanks to all of you who came and listened tonight and joined us. Thanks to you for your continued commitment to the Project Baseline Health Study. We look forward, all of us, the staff, the clinicians, the physicians, look forward to seeing you again on campus as soon as possible. And finally, thank you. Thank you for your commitment to this important project. And let's make it, as I said before, a phenomenal success to recognize the important contributions that Sam Gambier made before his tragic and untimely passing. Take care of yourselves, stay safe, and be well. Have a great rest of your evening.